What if the stress you feel at work isn't, just in your head?
Right.
Like, what if it's actually physically picking the locks of your gut, escaping into your bloodstream, and building a microscopic hormone factory inside a tumor?
I mean, it honestly sounds like science fiction, but that is the exact biological chain reaction we're looking at today. We are unpacking a truly groundbreaking paper from the journal Cancer Cell that completely redefines how we view the relationship between our minds and cancer progression.
Yeah, and welcome to today's deep dive, by the way.
Yeah.
Because we've all heard for years that chronic stress is bad for us, right?
Oh, absolutely.
And there's always been this sort of murky diagnostic link suggesting it can accelerate cancer. But the how has always been the missing piece. Like, how does a feeling of anxiety actually translate into accelerated tumor growth?
Right, the mechanism.
Exactly. And these researchers, they didn't just find a correlation, they mapped out this mind-blowing microscopic multi-species alliance hiding inside us.
They really did. And to understand how they uncovered this trans-kingdom alliance, we have to start with how they created chronic stress in the lab.
Okay, set the stage for us.
So they used a highly standardized model called CUMS. That stands for Chronic Unpredictable Mild Stress.
I feel like the word unpredictable is doing a lot of heavy lifting there.
It absolutely is.
Yeah.
Because if you expose a mouse to the same stressor every day at, say, noon, it eventually habituates. It learns the routine.
Right, the surprise wears off.
Exactly. So instead, the researchers took adult mice and exposed them to 2 random mild stressors a day for 21 days straight.
Wow. What kind of stressors are we talking about here?
We're talking about things like 12 hours of food or water deprivation, being restrained for 30 minutes, dealing with wet bedding.
Ugh, gross.
Yeah, a tilted cage, or even just having continuous white noise playing. They never knew what was coming or when.
It's literally the biological equivalent of a terrible, unpredictable month at work. Where your boss keeps changing the deadlines, your car breaks down, the AC goes out.
That's a perfect analogy. And to prove this was translating into genuine physiological anxiety, they used an elevated plus maze.
Okay, what is that?
Picture a cross-shaped platform raised high off the ground. 2 arms of the cross have high enclosed walls, and the other 2 arms are completely open and exposed to a sheer drop.
Oh, I see.
Naturally, mice want to explore. But the stressed mice, they showed this profound, terrified aversion to the open arms.
They just huddled in the safe, enclosed spaces.
Exactly. And when the researchers looked at their blood, they found the physical footprint of that fear, like massively elevated levels of corticosterone.
Which is a primary stress hormone in mice, right? Sort of similar to cortisol in humans.
Spot on. That flood of corticosterone is our starting point here. But hormones don't just float aimlessly. They're chemical keys looking for specific cellular locks. And for corticosterone, that lock is a receptor called NR3C1, the glucocorticoid receptor.
Okay, NR3C1. And it turns out the cells lining your gut, your intestinal epithelial cells, are basically covered in these locks.
They are.
So the stress hormone travels from the brain and binds directly to the gut lining. What actually happens the moment that key turns in the lock?
Well, the researchers observed a massive downregulation of 2 highly specific critical genes, LGR5 and Lys1. And this is really where the physical damage begins.
Break those down for me.
Sure. LGR5 is a genetic marker for intestinal stem cells. These are basically the cellular masons responsible for constantly regenerating the gut lining.
Okay.
And Lys1 is a marker for Paneth cells. Those sit at the bottom of your intestinal crypts and secrete antimicrobial peptides.
Okay, let's unpack this with an analogy. Think of the gut lining as a fortress wall. By suppressing those specific genes, the stress hormones are essentially firing the fortress's repair crew, the stem cells, and putting the defensive archers, the Paneth cells, to sleep.
That's exactly what's happening.
But wait, that makes absolutely no evolutionary sense. The gut barrier protects us from literally 1 trillion bacteria. Why would the body's own stress response intentionally sabotage its own defenses?
It seems like a catastrophic evolutionary glitch, right?
Yeah, exactly.
But what's fascinating here is the paradox of metabolic triage. From an evolutionary perspective, acute stress, like a sudden spike in adrenaline and corticosterone, means you are running from a predator.
Right, you need energy to run right now.
Exactly. But chronic stress, like this 21-day unpredictable model, signals to your biology that you are in a protracted, unending crisis. Resources are scarce.
So the body is forced to make a terrible trade-off.
Precisely. It reprioritizes immediate moment-to-moment survival over long-term structural maintenance. Rebuilding a gut wall requires enormous metabolic energy.
I mean, that makes sense.
Yeah, under chronic stress, the body essentially says, we can't afford to pay the cellular masons right now. We need all energy diverted to immediate survival.
And the tragic result is a neglected, compromised fortress wall.
Exactly. And they actually proved this fortress wall was falling apart using a really clever technique called the FITC dextrin assay.
The glowing dye,
Yeah.
right?
Think of it like swallowing a harmless, glowing fluorescent dye. In a healthy mouse with an intact gut wall, that dye stays inside the digestive tract and eventually passes through.
But in the stressed mice?
In the stressed mice, the glowing dye leaked straight out of the intestines and illuminated their bloodstream.
Wow. It is absolute visual proof of massive gut permeability, or, what we commonly call leaky gut.
Yes.
So let's do a quick recap of this first segment for you listening. Brain stress creates corticosterone that hits the NR3C1 receptors in the gut, which drops stem cells and Paneth cells, causing a leaky gut wall. Have you ever noticed your digestion going completely off the rails during a stressful month at work?
I know I have.
Right. Now, you know the cellular reason why, but if a glowing dye is leaking out of the gut, that means the trillions of bacteria living inside the gut are leaking out too.
That is the terrifying reality. A breached wall means a prison break. But when the researchers started hunting through the blood and the tumors of these mice, they didn't just find a random assortment of gut bacteria.
Right, it wasn't just a free-for-all.
Exactly. They found a highly specific culprit, a pathobiont called Enterococcus gallinarum, or egg.
So egg makes a run for it, but it doesn't just float around the blood aimlessly. It actively seeks out the tumor.
It does.
And when the researchers pulled the egg out of the tumors and ran whole genome sequencing on it, they realized something crazy. The egg inside the tumor wasn't genetically identical to the egg living peacefully in the gut.
Right. The tumor-bound egg, which they started calling egg-tum, had acquired a suite of specific genetic mutations.
So the thief didn't just escape the castle, it picked the locks to the new hideout.
That's a great way to put it. They isolated mutations in 3 vital genes.
Yeah.
Share, Magaya, and Ye.
Okay, let's break down what those specific genes actually do. Start with Share.
So Share is essentially a bacterial GPS system. It's a methyltransferase enzyme that controls chemotaxis.
Which means it allows the bacteria to sense chemical gradients in the bloodstream.
Exactly. It literally steers them toward the unique chemical environment of a tumor.
Unbelievable. Okay, what about Mengoga?
Mengoga is a transcription factor that acts kind of like a cloaking device. It regulates virulence, helping the bacteria form a physical shield to evade the host's immune cells while they're traveling in the blood.
And the last one, YH.
That's a transmembrane protein. It helps the bacteria adapt to toxic, nutrient-deprived environments.
Which is exactly the kind of harsh condition you find inside a rapidly growing tumor.
Exactly.
Okay, but this raises a major chicken or egg question about the causality here.
Yeah.
Did the corticosterone stress hormones physically cause these bacteria to mutate? Or did the stress just lower the drawbridge, allowing pre-mutated hyperadaptable rogue bacteria to escape?
It's a great question. And the evidence strongly points to the latter. It's a classic case of natural selection happening within the body. Well, within any massive bacterial population in your gut, there are always minor genetic variations. The stress-induced leaky gut creates the opportunity, but the human bloodstream is an incredibly hostile environment for gut bacteria.
Right, most of them would just die in transit.
Exactly.
Oh.
Only the single-celled organisms already equipped with that upgraded shared GPS and the Mengoga armor could survive the journey through the blood and successfully colonize the tumor.
Wow. It's literally survival of the fittest happening in real time inside your veins.
It is.
So let's hit our second checkpoint. Leaky gut lets egg escape. Egg mutates those 3 genes. Share, H, ego, and colonizes the tumor. If you were a bacterium, what kind of armor would you need to build to survive a trip through the human bloodstream?
Well, apparently exactly that kind.
Right. But here's where it gets really interesting. Once egg arrives at the tumor, it doesn't just sit there. This is where the study goes from fascinating to deeply unsettling. The bacteria bring a hidden passenger.
They do. Through that same genetic sequencing, the researchers found that egg-cum harbors prophages integrated directly into its DNA.
Prophages?
Yes. Specifically, they identified 2 viral sequences, which they simply called phage 1 and phage 2.
Let me make sure I have this straight. A bacteriophage is a virus that specifically infects bacteria, right?
Right.
So we are looking at a biological Russian nesting doll here, or a Trojan horse. A virus hiding inside a mutated bacterium hiding inside a tumor.
That is exactly what it is.
Yeah.
And because of those unique mutations we just discussed, this specific strain of egg is highly unstable. It is prone to activating what is called the involutic cycle.
And the lytic cycle is when the sleeping virus awakens, hijacks the bacteria's internal machinery to multiply, and ultimately explodes the bacterial cell from the inside out.
Precisely. And the researchers proved this using a mitomycin C assay.
What does mitomycin C do?
It's essentially a chemical trigger that forces dormant phages into the lytic cycle. When they exposed the normal gut egg to this chemical, nothing much happened. But when they exposed the mutated tumor-derived egg, it formed visible plaques in the petri dish. The bacteria burst open, spilling massive amounts of phage 2 viral DNA directly into the surrounding environment.
So now we have a tumor microenvironment flooded with naked viral DNA. The body has to react to that, doesn't it? Like, who is detecting this viral spill?
That brings us to a specific receptor called toll-like receptor 9, or TLR9. These receptors are designed to detect unmethylated DNA, which is a hallmark of viruses and bacteria.
Right.
And inside the tumor, the cells that are absolutely covered in these TLR9 receptors are called cancer-associated fibroblasts, or CFs.
I always think of CCAFs as cancerous architect friends, like they're the specialized cells that build the physical scaffolding, the collagen matrix that holds the tumor together.
Yes, they are the architects of the tumor microenvironment. Yeah. But when their TLR9 receptors detect this specific phage DNA, the architects actually change their blueprints.
What do you mean?
The researchers observed the CFs rapidly upregulating the expression of enzymes called STAR and CYP11A1.
Wait, I know those enzymes. STAR and CYP11A1 are the heavy molecular machinery required to synthesize steroid hormones. Are you saying the CCAFs start producing corticosterone locally?
That is exactly what is happening.
The very same stress hormone the brain was making, which started this whole cascade, is now being manufactured inside the tumor.
Yep. If we connect this to the bigger picture, it's terrifyingly brilliant.
I mean, a bacterial virus is essentially blackmailing your own cellular architects into building a localized stress hormone factory. But why? What is the tumor's biological motive for doing this?
It is the ultimate survival mechanism for the cancer. By manufacturing local glucocorticoids, the CCAFs create a highly concentrated, microscopic smokescreen of stress hormones right at the site of the tumor.
And we know that stress hormones are some of the most potent immunosuppressants in the biological world.
Exactly.
Let's do another quick recap. Egg in the tumor releases phage 2 DNA. That DNA hits TLR9 receptors on CCAFs, and those CCAFs start making local corticosterone. Isn't it wild that a bacterial virus is basically blackmailing your own cells into making stress hormones?
That's mind-blowing.
So the tumor is building a force field. Who are the primary victims of this localized hormone smokescreen?
The primary victims are a specific population of immune cells called germinal center, or GC, B cells.
B cells, those are like the heavy artillery of your immune system, right?
They are. In the context of cancer, their job is to mature and produce highly specific anti-tumor IgG antibodies.
Which physically tag cancer cells for destruction by the rest of the immune system.
Right, but manufacturing millions of specific antibodies requires an astronomical amount of cellular energy.
Of course it does.
To mature and deploy those weapons, a B cell has to undergo a massive metabolic switch. It has to transition from a process called glycolysis to a much more complex process called oxidative phosphorylation, or OXPHOS.
Okay, let's visualize that. Glycolysis is kind of like driving a sports car in first gear. It gets you moving quickly, it burns glucose fast, but it's incredibly inefficient and you can't sustain a high speed.
That's a great analogy.
And XPHOS, which happens deep inside the mitochondria, is like shifting into fifth gear to cruise down the highway. It produces massive, sustainable amounts of energy.
Exactly. And the researchers actually watched this metabolic failure happen in real time using a seahorse assay.
A seahorse assay?
Yeah, it's an incredible piece of technology that literally measures the oxygen consumption of live cells. So you can see exactly how they are breathing and producing energy.
Wow. And what did the cellular breathing show them?
It showed that the local corticosterone produced by the tumor's architects binds directly to the NR3C1 receptors on the B cells.
The same receptors from the gut.
The exact same ones. And when that happens, it acts like a physical governor on the B cell's engine. It completely blocks the cell's ability to shift into IXPHOS.
So the sports car is forced to drive on the highway in first gear. The B cell just burns out.
It does. It can't mature. It doesn't have the energy to deploy the anti-tumor antibodies, and the tumor gets a free pass to grow unimpeded.
Man, and how did they prove that was the exact cause?
Well, they performed an experiment where they genetically deleted the NR3C1 receptor specifically on the B cells.
Meaning the B cells were essentially blind to the stress hormones.
Exactly. They also did another trial using a drug called anti-CD20 to deplete B cells entirely. In both cases, the tumor-accelerating effects of the chronic stress completely vanished.
Wow, that is the smoking gun. I mean, the mice were still stressed, their guts were still leaky, the mutant bacteria still escaped, and the viral DNA was still blackmailing the fibroblasts. But without the ability to sabotage the B cells, the tumor didn't get its growth advantage.
Yep. The entire deadly circuit hinges on keeping those B cells stuck in first gear.
Okay, checkpoint 4. Local corticosterone means B cells can't use OXPHOS, which means no anti-tumor IgG, which means the tumor grows faster. Think about a time you got sick right after a stressful deadline. Could your B cells have been stuck in first gear?
It's highly likely.
Which forces us to ask the ultimate question. Is this complex, multi-species heist just a fascinating phenomenon in laboratory mice, or does this trans-kingdom circuit exist in humans?
That is always the million-dollar question with mouse models, isn't it?
Always.
Yeah.
Did they look at human data?
They absolutely did, and the parallels are striking. They examined human colorectal tumors provided by the National Cancer Institute.
And what did they find?
Just like in the mice, the human tumors showed significantly higher local levels of corticosterone compared to the healthy adjacent colon tissue. And inversely, those high-stress tumors had significantly fewer mature germinal center B cells.
Okay, so the local hormone factory and the burnt-out B cells are definitely present in human cancers. But did they find the bacterial delivery vehicle and the viral payload?
They did. They cultured live bacteria from fresh human colorectal tumors. Now, they didn't find the exact same Enterococcus strain, but they found something functioning identically, a strain of Klebsiella pneumonia, which they named KPT8b. And when they ran the genome sequencing on KPT8b, they found it was loaded with lytic phages.
So it's the exact same playbook, just a different microbial quarterback.
Exactly. And to prove it, they took the culture supernatant from this human KPT8b, which is basically a soup full of viral phage DNA, and applied it directly to human lung and pancreatic CFs in a petri dish. The human fibroblasts immediately started pumping out corticosterone. It is a highly conserved mechanism across different bacteria and different types of human cancers.
You just mentioned lung and pancreatic fibroblasts. The sources also noted this wasn't just isolated to gastrointestinal cancers, right? They found evidence of this in the brain.
Yes, they did. They reanalyzed a massive metagenomic dataset of human gliomas and metastatic brain tumors. And even across the blood-brain barrier, they found a statistically significant enrichment of phage DNA in the brain tumors compared to control tissues.
That is incredible.
It really seems like the phage TLR9 axis is a generalized, fundamental mechanism that tumors exploit, regardless of their location in the body.
Okay, so if we have the entire map of this circuit, from the stressed brain to the leaky gut to the mutating bacteria to the viral payload to the fibroblast hormone factory, and finally the burnt-out B cell, how do we break it?
Well, the beauty of having a map is knowing exactly where to set up a roadblock. The paper outlines two very clear, highly effective interventions.
What's the first one?
First, you can simply target the delivery vehicle. When the researchers injected ampicillin, a common broad-spectrum antibiotic, directly into the mouse tumors, it killed the egg bacteria.
Let me guess. No bacteria meant no viral release.
Exactly. The localized corticosterone dropped, the B cells recovered their energy, and tumor growth slowed down.
But relying on antibiotics inside a tumor can be tricky, right? What was the second intervention?
They targeted the TLR9 receptors on the fibroblasts using an antagonist drug called ODN-2088.
ODN-2088, how does that work?
This drug essentially blindfolds the CCAFs.
Yeah.
When they injected it into the tumors, the CFs could no longer see the viral DNA. The blackmail stopped.
So the local hormone factory shut down, the B cells shifted back into fifth gear, and robust anti-tumor immunity was restored.
Spot on.
Let's do our final recap. Human colon and brain tumors show this exact same viral DNA hormone factory activity, and we can potentially block it using targeted antibiotics or TLR9 blockers. If we can map this out, what other microscopic alliances are hiding in plain sight?
It's an exciting time.
Yeah.
We are entering an era of medicine where we are no longer just treating a single mass of rogue human cells. We are treating entire interconnected microscopic ecosystems.
Let's synthesize this whole incredible journey because it really is a masterpiece of biological detective work.
It really is.
We started with chronic, unpredictable stress in the brain. That flooded the systemic circulation with corticosterone, which shut down the cellular masons and archers in the gut, causing a leaky barrier. A mutant hyperadaptable bacterium equipped with upgraded GPS and armor escaped the gut and navigated to the tumor. Once inside, it burst open, releasing a bacterial virus. That viral DNA tricked the tumor's architects into building a local stress hormone factory, and that local stress hormone acted as a physical governor on our B cells, stopping them from shifting into high gear to fight the cancer.
It is a phenomenal demonstration of how interconnected our systems are. And this raises an important question, something profound and almost unsettling to ponder.
What's that?
If our immune system readily sacrifices its long-term defense, our B cells, to deal with the perceived immediate danger of chronic stress, and our own microbiome exploits this vulnerability to build localized hormone factories. What else is happening right now?
Oh, wow. It makes you look at modern life completely differently. Things like chronic sleep deprivation or, the constant low-grade digital anxiety of being tied to our phones and news cycles 24/7.
Exactly. We evolved to handle a tiger chasing us for 5 minutes. We did not evolve to handle a physiological alarm bell ringing for 5 years. Are those modern chronic lifestyles actively creating new, undocumented microscopic alliances inside our bodies today?
It brings us right back to our original premise. The old diagnostic expectation of a simple, clean, broken bone on an X-ray is entirely obsolete here. The landscape is murky because we aren't just one single organism. We are a walking, breathing, highly reactive ecosystem. But as this groundbreaking paper shows, even the muddiest waters can be mapped.
Perfectly said.
Thank you for coming along on this deep dive with us. Keep questioning the hidden connections in your own biology, and we'll catch you next time.